This is our presentation about tissue-specific transcriptomic changes in response to sleep deprivation, and it's based off Figure 2 by the paper given to us. My name is Sierra Follett.
My name is Anita Valdovinos.
I'm Cindy Vasquez.
And I'm Madeline Jewell.
So some terms that we wanted to go over before we started getting into our figure were differentially expressed genes, which are just genes that are changed in a significant amount. VLM, which is vastus lateralis muscle. SAT, subcutaneous adipose tissue. GSEA, gene set enrichment analysis. And KEGG, which is Kyoto Encyclopedia of Genes and Genomes.
So chronic sleep loss has been associated with metabolic pathologies such as obesity, metabolic syndrome, type 2 diabetes, and increased weight gain. It's proposed that sleep loss can cause a circadian misalignment, which could negatively affect tissue-specific metabolic pathways in adipose and skeletal tissues. Acute sleep loss could promote sarcopenia, which is the loss of muscle tissue, reduce respiratory exchange ratio, and promote adverse tissue-specific catabolism and anabolism.
So something that's talked about a lot in this paper is upregulation and downregulation of gene transcripts, and this is an example of gene expression alteration. So the way that they're able to see this and understand that it's happening is by RNA sequencing, so they can actually see transcription levels and see how much they're changing. And upregulation and downregulation is usually in response to another influence outside of the body or inside the body, but in a different system. So an example of this is sleep loss, which is what we're talking about, but other things can be immune influence, temperature stress, drugs, depending on the certain mechanism of action, and other things can cause these upregulations or downregulations. And what happens when genes or cellular components are upregulated or downregulated is the activity of that corresponding part or function that is related to what is being regulated. That will be either working less or working more. And this picture on the slide is an example of receptor upregulation and downregulation. It's just kind of a simplified representation of what happens in this process.
So these researchers used different sequencing analysis methods in their experiments for figure 2. For figures 2A, 2B, and 2C, they used RNA sequencing and compared this to an Illumina chip database for analysis of mRNA data. While for figure 2D, they used gene set expression analysis to find what genes in our tissues of interest had upregulation or downregulation of a specific gene transcript. So in figure 2, these researchers wanted to assess genome-wide gene expression changes that followed acute sleep loss in humans. And in this experiment, it was young Caucasian males. And in order to perform transcriptomic RNA sequencing and analysis of RNA, the RNA had to be isolated from the corresponding skeletal muscle and adipose tissue samples. RNA sequencing is a mapping technique that utilizes next generation sequencing to find the presence and quantity of RNA in a biological sample at any specific developmental stage or physiological condition. And it's used to map transcribed regions and gene expression and allows us to interpret the functional elements of the genome. With RNA sequencing, we're able to look at alternative gene spliced transcript, post-transcriptional modifications, gene fusion, mutations, and changes in gene expression over time, or differences in gene expression of different groups. But in order to compare the RNA of these tissues to Illumina chip database, the RNA sequence library has to be prepped first. And this involves mRNA being isolated from their tissues of interest, which in this case are skeletal muscle and adipose tissue. And then the long mRNA strand has to be broken up into smaller fragments because the Illumina sequencing machine will only read small transcripts. And these fragments are then converted into double-stranded cDNA because cDNA is more stable than RNA, and it's also easily amplified and modified. And once the RNA has been converted into cDNA, sequencing adapters are added to the ends of the fragments, which is important because this allows the Illumina analyzing machine to recognize individual fragments. And after the adapters have been added to the fragments, the PCR can be used to amplify the library. And the RNA sequence library is ready for comparison to the Illumina chip database. And the analysis of the mRNA data from the adipose in muscle tissue samples of muscle and adipose tissue samples will reveal any genome-wide gene expression changes following acute sleep loss. And in this picture, it shows just the basics of it. So here's the long mRNA and the smaller fragments that are then used to reference the genome, which was the Illumina genome database. And for figure 2D, they use gene set enrichment analysis, which is a computational method that is used to identify classes of genes that are overrepresented in large sets of genes. And this analysis determines whether a previously defined set of genes show statistically significant differences between two biological states. So this method derives its power from focusing on gene sets, which are groups of genes that share common biological function, chromosomal location, or regulation. And the goal of GSEA is to determine whether members of a gene set tend to occur toward the top of the gene list or the bottom of the gene list. And the top and the bottom of the list correspond to the largest differences in gene expression between two cell types. If a gene set falls at the top or the bottom of this list, then it's thought to be related to phenotypic differences, which is why these researchers use this method to find if acute sleep loss caused a phenotypic difference in gene expression of the two tissue samples. And this is just kind of a figure that shows, this is how they're mapped, the gene sets, and this is a correlation. So here's like the top or the bottom or vice versa.
So here's an image where the vastus lateralis muscle is located. And for the figure 2A, we used, the experiment was used a qPCR, which was used to analyze gene expression. The RNA sequencing was carried out for the genome-wide analysis of transcription. Illumina was used to prepare libraries for the sequencing, and the GSA was used for differential gene expression using the CAKE database. And you'll see in the next figure of what the results showed. So here, in order to visualize the gene expression data from the RNA sequencing was through the use of heat maps. Each row represents a gene and each column represents a sample. The colors on the left image represent a gradient. The bright red represents upregulated genes and the blue represents downregulated genes. The image on the right-hand side has a different color gradient. The blue means that the wake expression is 2 times as much as sleep expression, and the red means that the wake expression is 2 times less as sleep expression. The white represents unchanged or no differences between the two conditions. An approach of interpreting the gene expression data was through the GSEA, which helps find out if the differentially expressed genes are associated with certain biological processes. The data showed an altered expression of 117 mRNA transcripts in skeletal muscle, 19 upregulated and 98 downregulated. And one of them was TEX-NIP, which was upregulated, which tends to be regulated with upregulated with pre-diabetics or diabetic patients. Other upregulated genes are inflammation related and with mitochondrial energy metabolism. Another one found was HSF1, which was downregulated, which is consistent with upregulated HSP, which are heat shock protein levels. HSP helps stabilize glucocorticoid receptors that have been implicated in skeletal muscle atrophy, affecting type 2 fibers. This suggests that glucocorticoids may be involved in acute catabolic effects on skeletal muscle following sleep loss. Downregulation in the skeletal muscle has been positively correlated with insulin sensitivity, further suggesting that sleep loss may promote catabolic strain on several immune, injury, and stress-related genes.
So figure 2b follows the same type of representation and the same heat map system of showing their results. And this one is focused on subcutaneous adipose tissue. And this type of adipose tissue is normally known as energy storage in the form of fat, but adipose tissue is also involved in hormone production. So this is an important part of the body to look at in terms of sleep loss and how it's affected, because when expression levels in the adipose tissue of certain genes are altered, it has been linked to insulin resistance. And in this this representation of the data shows that 96 mRNA transcripts were altered significantly. So 59 were upregulated and 37 were downregulated. And among these that were upregulated were CD14 and TLR4, which are both involved in glycolysis and toll-like receptor signaling. And this is important because, like I said, they can be correlated to insulin sensitivity. And the upregulation shows that it could be linked to the body favoring adipose tissue in the circumstances of sleep loss, while the figure beforehand, as Anita discussed, showed that the catabolic activity could be not favoring the skeletal tissue or the muscle tissue. So that basically simple words would be favoring fat, losing muscle.
For figure 2C, we see the relation between significantly expressed genes and of skeletal muscle and adipose tissue, in which the researchers found that there's actually almost no overlap between VLM and SAT, which VLM is the muscle and SAT is the adipose tissue. For the mRNA transcripts that were differentially expressed. This Venn diagram shows the genome-wide gene expression changes caused by acute sleep loss that were found in this study. So from figure 2A and figure 2B, we learned that the authors performed transcriptomic RNA sequencing analysis of total RNA isolated from the corresponding skeletal muscle and adipose tissue samples. And they found that 117 mRNA transcripts in skeletal muscle, or VLM, as it appears here in the green and purple, had altered gene expression, in which 98 out of the 117 had downregulation of gene expression, and 19 of the 117 had upregulation. They also found that only 96 mRNA transcribed in subcutaneous adipose tissue, or this is the orange and the red tissues, had altered expression, in which 59 out of the 96 mRNA transcripts had upregulation of gene expression and 37 had downregulation in this tissue. As you can see, there was only one instance here where there's overlap for significantly regulated genes between SAT and VLM, and overall, there was no overlap between the two tissues for mRNA transcripts that were differentially expressed, which supports that there is tissue specificity of the response to acute sleep loss in human metabolic tissues.
So for figure 2D, the KEGG pathways were analyzed using the statistical program GAGE to produce a GSE-A showing the most significant genetic regulatory pathways that were affected by acute sleep loss. The analysis is separated into its two respective tissues and is flanked by parallel columns. On the left is the corresponding Q value for each entry with the complete list of all up and down regulated pathways in each tissue. And on the right is the scale. So a Q value is a p-value that has been adjusted for various false discoveries. When we use a p-value to determine significance, we typically set the alpha value to 0.05. This means that there's often a less than 5% chance of the data being significant just by chance. In a large data set like this one, there's bound to be some individual data that yields a false discovery. This discrepancy is accounted for by using a Q value with a false discovery rate of less than 0.05. The most significant trends will be at the top and the least significant trends are at the bottom. So for skeletal muscle, the most significant regulatory pathways that were affected by sleep loss were the downregulated genes associated with ribosomes and oxidative phosphorylation. Oxidative phosphorylation is the final step of cellular respiration and is the critical component in determining aerobic respiration from anaerobic respiration. The disruption of oxidative phosphorylation in skeletal tissue goes hand in hand with further disruption of the body's circadian rhythm, which can lead to a cascading effect on the transcriptome. Other pathways that were affected were those for the translational and cellular protein targeting processes. For example, energy demanding and stress sensitive biological processes. So this further suggests that sleep loss promotes catabolic stress, including several immune injury and stress related genes, such as NMT, CXCR2, LRRK2, and FCGR3B. For the adipose tissue, it was the opposite. Oxidative phosphorylation was upregulated. Additional upregulated pathways included glycolysis and related functions, as well as toll-like receptor signaling. By upregulating the TLR genes like CD14 and TLR4, it can throw off balances in adipose tissue when it comes to insulin sensitivity. Again, suggesting that it increases fat and decreases losing fat because it increases energy storage. In conclusion, the results from Figure 2 can be summarized into 4 distinct conclusions. So the effect of acute sleep loss on the body affects different tissues in different ways. This was shown by Figure 2C. In this figure, it was demonstrated that there is a lack of overlap between mRNA transcript expressions for both tissues in the study. The second is that specific regulatory pathways can experience an increase or decrease depending on the tissue samples. So in Figure 2D, it's shown that the VLM tissues experience downregulation with vital functions like oxidative phosphorylation and ribosomal functions, while for the adipose tissue, functions like glycolysis and TLR signaling were upregulated. Figure 2A displays the vastus lateralis muscle tissue and all of the 117 mRNA transcripts that undergo altered expression. Specific genes and specific functions include the upregulation of gene TXNIP, which can be linked to inflammation, while the downregulation of gene HSF1 and others can increase likelihood for immunological injury or stress. Finally, Figure 2B displays the adipose tissue samples and the 96 mRNA transcripts that underwent an altered expression. The upregulation of specific genes like CD14 and TLR4 may be correlated with insulin sensitivity in adipose tissue.